So today I will be talking about the drug discovery and development from chemicals to treatments. The purpose of this presentation is to explain how modern drugs development works and to demonstrate why sustained investment in this research is important for advancing science and supporting commercial innovation. In this presentation, I will discuss the drug discovery and development and involves transforming chemicals concept into medicines as they are being used to treat, prevent, or control diseases. Medicines are not created by any chance as they are being produced through years of work on through years of scientific uh, investigations, laboratory testing, and strict safety evaluation. Pharmaceutical science brings together chemistry, biology, and medicines with the aim to improve the human health. As scientists and a pharmacist, our role is to ensure that medicines are effective, which means that they are achieve the intended therapeutic effect safely, uh, as they do not cause any unacceptable harms and suitable, which means that they can be produced, stored, and used correctly by patient. So as we can see, the history of drug discovery diagram, as it's showing how drugs discovery has changed over time, as it begins on the herbal drugs, then moved to vitamins, antibiotics, uh, the use of IT, breaking through ethology, discovery of DNA, and so on. Later on, science and technology improved drug research, and today, computers and automation help develop medicines much more faster. What's medicine? A medicine is a chemical substance that produces a biological effect in the body that support health and improve the quality of life. Medicines, uh, medicine actions can be classified into three main categories. The first category is curative medicine. Curative medicine it's designed to cure or eliminate diseases, uh, and a common example is antibiotics, which are used to treat bacterial infections. The second type of medicines is preventive medicine. Preventive medicines can help stop disease before it develops, and vaccines are a clear example that they can protect the body against any future infections. And the third type of medicine is symptom relieving medicine. So symptom relieving medicine, which reduce the discomfort such as pain, fever, or inflammation. These drugs do not cure the disease itself, but help patient feel more comfortable. Medicines function by interacting with specific biological target in the body, including receptors, enzymes, or DNA. When drug binds to its target, may activate or block biological pathway, leading to intent therapeutic effect. As we can see here in our diagram, it shows how drug works by binding protein as the drug fit into a specific vein site on the protein, like a key fitting into a lock. As when the drug binds uh, forms a drug protein complex which can block or change the protein action. As we can see here, so the role of placebo in medicine research. So a placebo is an inactive substance that is designed to look identical to a real medicine as it does not contain any active ingredients. And patients do not know whether they are receiving the real drug or the placebo. The placebo are very important medical search in medical research, sorry, as they help scientists fairly to compare new medicine with current treatment during clinical trials, as this show if the new medicine are working or not. Also, placebo helps scientists understand how does it affect the mind health, as some people feel better simply because they believe they are receiving the treatment, which is called the placebo effect, as they just feel that they are getting the treatment while they're not actually getting the treatment. As we can see here, there's a diagram uh, showing two people. One of them is taking the placebo, which is an identical look of medicines but does not have any active ingredient. Uh, as they feel better, the treatment makes the brain respond and feel better. While on the other hand, the real drug causes improvement because it contains the active ingredient. Therefore, the key idea is that belief and expectation can affect how the brain and body feel without a real medicine. Overall, a placebo show why careful testing is needed before medicine is approved. Using a placebo helps make sure new medicine are safe, effective, and truly helpful for patients. So the therapeutic window. The therapeutic window refers to the dose of medicines that work well and at the same time does not cause any harm to the patient. The amount given must be enough to be effective, but at the same time does not harm the body, does not become dangerous. As medicines that have wide range of therapeutics are considered safer because the correct dose is easier to manage. As we can see here in the uh, line graph, like the diagram shows a therapeutic window of the drug shown. The low dose has no effect, as we can see here, and as increasing the dose, the effect is increased, and then uh, the middle range is best and safe uh, to use medicines, which is effective and at the same time it's not dangerous. While at the high dose, as we can see here, has a maximum effect range, which hits the plateau and it's not effective anymore, and it becomes dangerous to take the medicine than not taking it. And the key idea is that we've got to understand that there is an optimal dose range where the drug work best and safe. Drug tolerance. So, drug tolerance means that when medicines work less well, when they well, sorry. Drug tolerance means that when medicines work less well, when it being used again and again, over and over. So we've got to think about the long-term medicines. As tolerance develop, a high dose may be needed to get the same effect as before, as this can increase the chance of side effects and dependence. Understanding the tolerance is important when creating medicines meant for long-term use, as it helps ensure treatments remain safe and effective for the patient. So that's a diagram there showing the response curve of different drugs. So as we can see here, the x-axis is the concentration and the y-axis is the response. So in curve A, we can see it's most potent, which means it works at lowest dose, which is what researchers and drug discoveries are looking for. And as we can see here, in curve B, C, and D, showing the decreasing of potency, which means a higher dose needed, which researchers don't want. So the EC50 over here, it means the dose that produced 50% of the maximum response. So the key idea over here we've got to understand is that the drug with a low EC50 are more potent and what drug researchers are looking for. Side effects and side effects are unintended effects that occur when medicines affect the body in ways other than its intended action. They can happen for many reasons, such as drug interaction with multiple targets in the body, or individual reacting differently to the same medicine. So reducing side effect is a key goal in drug development as it helps make treatment safer, more comfortable, and more effective for patients. As we can see here, a diagram explains the behavior of the medicines. So divided into undesirable and desirable. So the undesirable behavior is reduced by a punishment or an extension, which means it's removing the reinforcement, and the desirable behavior, which is increased by a positive reinforcement, which is a reward. So the key idea is the behavior change based on the consequences. Molecular structure of medicines. Most medicines are organic chemicals that contain carbon as their main structural element. Medicines are medicines that belong to the same group often have similar core structure and functional parts, which is why they cause similar effect in the body. Even so, small changes in medicine chemical structure can greatly affect how well it works and how safe it is to be used, and how the body absorbs and uses it. Because of this, understanding chemical structure is a key part of developing effective and safe medicine. So that's a comparison of molecule structure similarity and variation. So small changes in drug structure can change how it works in the body, as aspirin and ibuprofen both reduce the pain and inflammation. But ibuprofen is easier on the stomach, while aspirin can cause stomach irritation. Penicillin antibiotic all share common structure that helps them kill bacteria, changing the side parts of the molecule. Scientists can be can be improve can improve the medicine. For example, amoxicillin dissolves better in the body, so it work well than taken by mouth. While methicillin has a larger side part that protect it from bacterial enzyme, helping it stay effective. So small changes in the functional group can change how drugs work in the body, as aspirin, naproxen, and ibuprofen are similar painkillers but different in their structure, effect, absorption, effectiveness, and side effect as it's shown in the diagram. Overall, changing parts of the drug structure helps make medicine safer, work better, and suit patient more. Functional group and drug effect. It explains in this slide. I will explain how functional group affect how functional group affect how medicines work in the body, as the functional group are parts of the molecule that control its chemical behavior and biological effect in the body. So by changing the functional group, scientists can improve the drug in different way, as they can help the drug be absorbed into the bloodstream more easily, and this group can slow down the body, breaks down, slow down, slow down the breakdown of the medicine in the body, of the drug in the body, which allowing it to stay active for a longer time. So modifying functional group is used to improve how well the drug works and how safe it is. This process, known as structure activity relationship or SAR optimization, helps scientists design medicines that are more effective and have fewer side effects. Improving drug delivery and safety. So on this slide, I will be talking about the how altering medicine's chemical structure can improve how it moves through the body and how safe it is for patients. These changes are key part of modern drug development and help create medicines that are easier to use and more effective. One improvement approach is the use of prodrugs. So prodrugs are medicines that are inactive or less active when first taken, but are changed into the active from inside the body, as this help limit the side effects, protect the drug as it travels through the body, and improve how well it reaches the target area. As the advantage of this improvement, another advantage of this improvement is the reducing harmful effect. So geometrical isomers, cis and trans. So some medicines exist as a geometrical isomers, meaning they have the same molecule formula but a different ar- arrangement of groups in spaces. So in cis isomers, the functional group are on the same side of the double bond or the ring, while in trans isomers functional group are on the opposite sides. Even though these molecules look similar, their different shapes mean they can interact with the body in different ways. One isomer may fit well into biological receptor and work effectively, while the other may be less active or causes unwanted side effects. This this is show why the understanding of geometrical isomers is important when developing medicines that are both safe and effective at the same time. So, detecting drugs in research and testing. Detecting drugs in research and testing as it describes how drugs are tested during research development as before medicines is approved for patient use must be checked carefully to make sure that it's safe, pure, and correctly identified. As there are five techniques that can be used, which are chromatography uh, is one of the main techniques used, such as uh, high pressure liquid chromatography and gas chromatography, as these methods separate the component of the mixture, allowing scientists to detect impurity and confirm the correct drug is present. So mass spectrometry is also a is commonly used uh, as it measures the mass of the molecule and their uh, fragments, helping scientists identify the drug's structure and detecting very small amount of substance. So other testing methods including infrared and nuclear magnetic uh, reson- resonances, resonance, which is NMR, and ultraviolet spectroscopy. Spectroscopy, sorry. These techniques provide details about the drug's structure, functional group, uh, identified, and purity. So overall, these testing methods ensure that medicines are high quality, accurately dosed, safe for patient to use, and suitable for approval and pharmaceutical use. So identifying potential new drug molecule. Identify potential new drug molecule is describes how drug molecule is discovered at the beginning of the drug development process, as scientists use several strategies to find compounds that could be useful as medicines. One method is that involves studying natural substance taken from plants, fungi, and microorganisms, as many of these show useful biological effect. Another method is focusing on changing and improving the existing drug to increase their effectiveness and lower their side effect. Sorry. As many possible drugs candidate are found, only few may safety and effectiveness standard needed to continue into clinical trials and highlighting how challenging drug development is. So, lead compounds and optimization. So, lead compounds is a substance that has shown a useful biological activity against target is and is chosen as the starting point of developing a new medicine. After lead compounds is selected, scientists refine its structure to improve performance, as these includes increasing its effectiveness. So smaller doses needed to are needed to be improving how specifically acts on the target and reducing unwanted side effect. So optimization also focuses on uh, pharmac- pharmacokinetics, which means that how the drug is absorbed, spread through the body, metabolism, and how it removes toxicity. This improvement helped the drug research to correct say this improvements helps the drug research to the correct site in the body as they stay active for the right amount of time and work safely. This stage requires a lot of time and resources as it's essential to for turning promising compound into safe and effective medicine. So the role of computers in drug design. Computers are new, very important in pharmaceutical pharmacy, pharmaceutical research. Sorry about this. As it helps scientists design and study drugs faster than using lab experiment alone. So computers are used to model how drugs fits into biological receptors as they help predict how well the drug might work before it's been made. As they are also used to predict toxicity and metabolism, which helps find safety problems only. This method, called computer aided drug design or CADD, and it helps developing safer and more effectiveness medicine. So the advantages of computational approaches is the main advantage of applying these techniques and during development as these methods allow researchers to analyze more efficiently and make well-informed decision at early stages of the drug discovery, as it saves a lot of time. One main advantage is that computers can quickly check many possible drugs compounds that they help identifying which ones are most likely to work, which saves a lot of time in early drug research. Another advantage is that reducing the need for lab testing and animal experimental, and again, it saves a lot of time. At this stage, at the end, using computational methods helps researchers focus on the best drug candidate, which speeds up development and helps produce safer and more effective medicine. Why continued finding matters? Continued finding matters is because making new medicines is complex process that needs knowledge from different areas such as chemistry, biology, and computing. Drug development also takes a long time as it can be as it can take many years from the first idea to approval, and many drugs fail before reaching the market. However, successful drugs can be greatly uh, improve patient health and bring financial benefits. Continued finding helps researchers work faster and focus on the best drug candidate and support proper testing, which leads to safer medicine as it also helps new drug reach patient more quickly. In conclusion, in conclusion, uh, we can understand that drugs are not random chemical as they are carefully designed to work with a specific target on the body. Therefore, each part of the drug structure is chosen to give the best benefit while reducing the harm and the side effect. So small changes in drug structure such as changing the group or length can greatly affect how it works, as these changes can make the drug more effective, less toxic and safer on the body to be used. So the modern drug development uses advanced chemistry and computer tools. These tools help scientists predict how drug will work and how the body processes them and whether they may be harmful, as this reduces the risk, saves time and lower the need for labs and animal testing. Finally, continued finding drugs is very important for drug research as helps improving drug design supports safety, testing, and speed up development, which allows safer and more effective medicines to reach patients sooner. And thank you very much for listening.